Hello, my name is Daniel Stone, and this is presentation 3 for the Dose Response Models group. I'm going to get the ball rolling with the Q&A from our second presentation. So without further ado. So the first question says, given that we now know how to test and model drug efficacy, can we apply the same technique to do potency control in the high volume manufacturing phase of the drug? This is a great question. It is a problem in the field and industry for failed phase 3 studies and programs due to inappropriate or incomplete knowledge of the dose response relationship, meaning efficacy and safety, at the end of the learning phase of the drug development, that there's often poor dose selection. This is just a difficult question, and it carries into the confirmatory phase 3 trials. And so there's this quote here: although the exact numbers are unknown, it is believed that the high attrition rate plaguing the pharmaceutical industry in phase 3 studies are due, at least in part, to inadequate dose selection for confirming safety and efficacy in the intended patient population. And it goes on from there. You guys can read it. But the idea here is that often the dose is it's ineffective, right? Either because it's too low or it's too high for whatever reason. And you see that there's another quote there with some numbers from Bayer Healthcare. And again, I don't want to just read this to you, but we'll go on here to the next slide. There was some teams that were set to work on this. And so the Pharmaceutical Innovation Steering Committee of the Pharmaceutical Research and Manufacturers of America, the PHRMA, they formed back, this is a while back, this was 2005, but they were looking into some of the different drivers and causes of the decreasing success rates in these drug development programs in the industry of pharmaceutical development. And what they did is they did this comparison study, and they were looking at some of these different things that were being developed, including novel adaptive and non-adaptive dose ranging methods. And one of those was the MCP mod. And so what they did is they presented their recommendations for industry and for regulatory agencies. And you can see the details from these white papers here. But that's kind of the idea, is that developing through phase 2 and into phase 3. And, this kind of carries on to the next question as well, which I'll come back to. But it's the idea of applying the MCP mod process in novel and adaptive ways. Now, I don't want to retread what was covered in the second presentation because Gerard did cover that, but I'll talk about some other ways that are applications and kind of novel ways to use this same process in other ways. So this is a great question. So as you can see here in the FDA evaluation, oh, you know what? Let me come back to that. There was a second question, and they don't overlap, but there is some relation. So the second question, how to solve the overfitting problem in model selection. And then the student gave an example. It's possible that the model we select fits very well with the current data, but once more data is collected, the model does not fit well. And so this kind of goes or continues on from the idea of some adaptive models and using the MCP mod process in some of these different ways. And so going back to the beginning here, the FDA evaluation of the MCP mod through, and there was these simulation studies that I just referenced. They compared the MCP mod to non-adaptive dosing methods. There was a ANOVA-based method that was included, a Bayesian approach, a non-parametric approach, and specifically the BMA is a strategy that's intermediate between the parametric modeling and non-parametric modeling, and it tries to avoid those specific dangers, right? The dangers of under or overfitting. And the idea is that, to avoid this pitfall in general. But the methods, the BMA and the MCP mod, they were comparable in the way that they control type 1 error rate and detect dose response, identify. These are some of the criteria that we went through in the first presentation as well, but the way they identify minimal clinical effective dose and so forth. But there was some proposed extensions of this. So you can see here Miller and Tanaka and Sampson extended the MCP mod approach to a flexible two-stage design. And this is actually really cool. It includes the possibility to perform adaptations to it, including adding or dropping doses under strict family-wise error rate control. And the goal, as always, to choose a model that fits the data well. And because this is not easy, the criterion for the model selection includes, or this is another way, I guess, to try to control for overfitting, but includes a penalty for the flexibility of the model. So as you guys if you ever do consider this process, the selection criteria should include that penalty. And this is a common statistical technique, right? But the idea is to say, okay, just because you have a model that fits the data extremely well, but it's not of any practical use, you wouldn't want that model. So you're looking for some like selection criteria that is going to account for that. A great example of this is an alternative approach to using the MCP mod in adaptive dose finding, and that is to use decision rules that are fine-tuned to specific trial considerations. And so as an example of this, in Mercier et al., they described a real case study of a two-stage design, and for decision making at the interim, the different hypothetical scenarios of observed dose response shape were set up. And for each scenario, a corresponding design for the second stage was pre-specified. At the interim analysis, you would then observe to which scenario the observed data correlate best, and the corresponding data is then selected for the second stage of the trial. The corresponding design is then selected for the second stage of the trial. And so an example of such a rule could be to include a lower dose at the interim if all the active doses are, at the interim are on the plateau of the dose response curve. And that was the case in the actual case study. So that would be interesting, if you wanted to press into this a little more and study some possible alternative approaches to using this method. So this was another question from presentation 2, and the answer to this one is pretty short and sweet, actually. The question is, what is the LDCL covariate in the trial referenced in part 2? And in the initial analysis, there was just a typo there on the slide. The LDLC was used as a covariate, and the LDLC is low density lipoprotein cholesterol. So that is the answer to that one. Now with this one, the question is simple enough, but finding the answer was a little bit problematic. Why was there a placebo dose in the safinamide case, but not in the antithrombotic case? And in this specific case, it states in the report that it was assumed that the lowest dose would be close to the placebo one, which I'm taking to mean that there was actually a placebo dose and it was just not reported in the data from the article that was cited in the last presentation. So I don't have a better answer at this point. I mean, obviously we all know there's sometimes reasons to not have a placebo case, usually some kind of ethical issue, or it's, a simple comparison or something. But yeah, it appears that in this one there actually was a placebo dose. This question is actually a great question, and I don't have an amazing answer for it, but I'm going to try to address it anyway, just because it's so central to the MCP mod process. So in the MCP step, if no model has a significant test, then no model is chosen from the reference set, and the modeling step is not performed. That's true. That's taken straight out of our other presentations. In this case, what is the next step? Is the new set of candidate models chosen, or a new set, or is a completely new approach taken with dose selection? And to tell the truth, either of those could be an answer, okay? And so I'm just going to try to go back and highlight a few things. The idea with this is to, we want to, well, okay, so let me say it this way. We consider the problem of determining the best contrast associated with a given model function, and that's, and in that sense, when the model is correct, it maximizes the chance of rejecting the associated null hypothesis. So it's maximizing the non-centrality parameter. Once we have the optimal contrast coefficients for each model in the possible candidate set, we evaluate the significance of the individual models in terms of the corresponding single contrast tests. And so each test translates into a decision procedure, whether the selected dose response curve is significant given the current data, and all this is controlling for family-wise error rate at a level of alpha. Now, a good candidate curve is chosen as one with a significant contrast test, given that its individual multiplicity adjusted p-value is less than alpha. And so if no candidate model is statistically significant, the MCP mod procedure does stop, indicating that a dose response signal cannot be established from the observed data. So this result does not necessarily mean that the compound or the thing that's being tested has no effect at all. In general, we will assess that there was not enough evidence to get statistical significance, and there could be possible reasons for this. Some of these have been stated in prior presentations, but they could be things like, small sample sizes, high variance. In addition, the initial candidate set plain and simply might be poorly chosen. And so, the candidate set or the candidate models just might not fit the true curve, perhaps like an umbrella shaped curve or something like that. Although in such cases, the compound, the thing that's being tested, has a pronounced effect and may be far from ineffective. So again, we're not necessarily saying that, that there's no effect. We're just saying that there's no statistical significance. So, and again, that's, suggested by an insignificant p-value. A couple more comments worth making here. The MCP mod has been shown to be sensitive to the initial model shapes chosen, which does give some weight to the idea of going back and revisiting your initial candidate set. Okay, and then also the FDA reviewers, when they approved the process, suggested not restricting the number of shapes to be included in the procedure. So again, perhaps having too limited a set of candidate your candidate set being too limited could also be a problem. And to give weight to that as well, though I don't have a slide for it here, in some of our research here for presentations 2 and 3, some of the adaptations that have been done with MCP mod, as well as other studies where they're taking this and applying this process in unique and novel ways, which I mentioned earlier, are giving weight to that as well, and showing that there are some other possible shapes that could and should be included in that candidate set. Having said that, in its publication, the method itself currently does not provide guidance on how the results should be interpreted or utilized when none of the candidate models are correct. So, whether it's going back and reviewing your candidate set, adding some different shapes, or trying a new process altogether, or, and as you can see here, perhaps the results could still be used. An example might be models meeting criteria that are less stringent than the pre-specified criteria. Could be considered, models for future studies, or, and then also, this is just repeating what I said earlier, so I won't rehash that second paragraph there. But the candidate set might have been poorly chosen. That is a possibility. And so, whether revising that candidate set or moving on to a different process is kind of left to the researchers. The final question I want to address was actually posed to us, but it could be a great question for all groups, and perhaps we're all going to hash this out in our own presentations and projects. But the question was, what was the most interesting thing that you learned by doing your presentation so far? And of course, that's a little bit different for each one of our presenters. But for me, I find the hybrid combination of approaches, the multiple comparisons and modeling in the MCP mod method fascinating. I've genuinely enjoyed seeing how it's been applied in different ways, the bridge that it provides with phase 2 and phase 3, the way it meets practical needs, in the industry, these well-known problems with dose finding studies, and even just choosing a correct dose and how difficult that can be. And then just seeing too, the statistical nature of this process is interesting, and seeing the way, that's why I got into the program to begin with anyway, is, the idea of using mathematics and statistics to solve real world problems. And that's what this does. It helps address known issues and provide, flexibility, adaptivity. And so that's been what I've enjoyed. And again, I'll pass it off to my other presenters. But that was a great question.